This content is AI generated for informational purposes only and should not be directly used for clinical decision-making.
Hello and welcome back to the Neurosurgery Hub podcast. I'm Dr. Eris, Senior Attending Neurosurgeon.
And I'm Dr. Alara, Research Fellow. Today we're looking at a fascinating new study from Epilepsia Open, published in 2026, titled sEEG-Guided Responsive Neurostimulation to Treat Neocortical Epilepsy, a multicenter retrospective study of the efficacy and safety of depth electrode-mediated neuromodulation.
This is a timely paper, Alara. For our residents and colleagues listening, we know that the RNS system has been a game changer for focal epilepsy since the pivotal trials. But much of our early experience was heavily weighted toward mesiotemporal lobe epilepsy. This study, led by Sina Sadiqzadeh and a large multicenter team, including folks from Stanford, Mount Sinai, Yale, and several other top-tier institutions, really zooms in on the neocortical population.
Exactly. And what's particularly interesting here is the methodology, specifically sEEG-Guided RNS. Historically, many neocortical RNS leads were placed using subdural strips or grids for localization, or perhaps just imaging. But this study looks at the efficacy and safety when we use stereo-electrocephalography, sEEG, to guide the placement of depth electrodes.
It's a natural evolution of the workflow, isn't it? We do the phase 2 monitoring with sEEG to find the seizure onset zone, and then we use those exact coordinates to place the therapeutic RNS leads. Let's dive into the core objectives of the study.
The primary goal was to evaluate the efficacy and safety of using depth electrodes for neuromodulation in patients with neocortical epilepsy. Being a multicenter retrospective study, it draws from a pretty diverse patient pool across institutions like Thomas Jefferson, Mayo Clinic, and Emory.
Right, and for the listeners, let's clarify why depth electrode-mediated neuromodulation in the neocortex is a bit of a shift. Traditionally, if you had a lateral frontal or parietal focus, many surgeons would gravitate toward a cortical strip electrode for RNS, but this study focuses on using depth leads instead. Ilara, why is that distinction important?
Well, depth electrodes allow us to target the seizure onset zone with incredible three-dimensional precision. If the sEEG shows that the bottleneck of the seizure network is 2 centimeters below the cortical surface, a strip electrode on the surface might not be as effective as a depth lead positioned right in that volume. The study really looks at how this depth-mediated approach performs in terms of seizure reduction.
And the multicenter aspect is crucial here. Neocortical epilepsy is heterogeneous. You're dealing with different lobes, different etiologies, maybe focal cortical dysplasia in one patient and post-traumatic scarring in another. By pooling data from Stanford, Penn State, OHSU, and others, they get a much more robust look at the safety profile.
Speaking of safety, that was one of the major pillars of this paper. When we talk about depth electrodes, residents often worry about the risk of hemorrhage or infection, especially when transitioning from a temporary sEEG study to a permanent implant.
Absolutely. Let's look at what they found. The study suggests that sEEG-guided depth electrode placement for RNS is safe. When you have a multi-center retrospective study like this, you're looking for any red flags in the surgical complications. Did they find any significant increases in adverse events compared to the standard RNS pivotal data?
No, the safety profile remained consistent with what we expect from the RNS system. The transition from the sEEG mapping phase to the RNS implantation phase was managed effectively across these centers. The incidence of serious complications related to the depth electrodes themselves was low, which is reassuring for surgeons who might be hesitant to put depth leads into non-mesial temporal targets.
That's a key clinical pearl. If you have the sEEG coordinates, you have a vetted target. You aren't guessing. You are placing the therapeutic lead exactly where the diagnostic lead saw the most action. Now, let's talk about efficacy. Efficacy of depth electrode-mediated neuromodulation. That's a mouthful, but it basically means, did the seizure stop?
Precisely. The study focused on seizure frequency reduction. While we've seen great numbers in mesial temporal cases, neocortical cases can sometimes be more challenging because the networks can be more diffuse. However, this study shows that by using sEEG to guide the RNS, the efficacy in these neocortical patients is quite compelling.
It's that precision again. If the sEEG identifies a focal neocortical onset and you can get those depth electrode contacts right in the sweet spot, the responsive stimulation is going to be much more targeted. The paper uses the term depth electrode-mediated neuromodulation to emphasize that the depth lead isn't just for the hippocampus anymore. It's a tool for the neocortical mantle.
I found the list of participating departments really impressive. You have neurosurgery and neurology departments from across the country, Dartmouth, Colorado, Washington University, and St. Louis. It shows a real consensus in the field that this sEEG to RNS workflow is becoming the standard of care for complex focal epilepsy.
It also highlights the team sport aspect of epilepsy surgery. You have Dr. Sadiqzadeh and the neurosurgery team at Stanford working with neurologists like Dr. Graber. You have the same thing happening at Mount Sinai with Dr. Gatan and Dr. Yu. For the residents listening, this study proves that your Phase 2 planning is essentially your RNS surgical plan.
Let's look at the patient selection mentioned. These were patients with neocortical epilepsy. In many cases, these are patients who might not have been candidates for a resection because the focus was in eloquent cortex, motor, sensory, or language areas.
That's the classic RNS indication. If you can't cut it out, you modulate it. And if you're going to modulate eloquent cortex, you want to be as minimally invasive as possible. A depth electrode has a much smaller footprint than a large subdural grid or even multiple strips.
And that brings us back to the title, sEEG-Guided. The precision of sEEG allows us to avoid major vasculature and eloquent structures while still getting the electrode exactly where it needs to be. The retrospective nature of the study allowed the authors to look at long-term outcomes across these multiple centers.
Ilara, based on the paper, what would you say to a resident who asks if they should use a strip or a depth lead for a neocortical RNS target?
I think this study provides strong evidence for the depth lead, provided you have high-quality sEEG data. The efficacy and safety demonstrated here suggests that depth electrode-mediated neuromodulation is not only feasible but highly effective for neocortical targets. It allows for a more targeted volume of stimulation.
Well said. It's about the volume of tissue activated. With a depth lead, you're stimulating a sphere around the electrode contacts, potentially capturing a seizure focus that sits in the sulcal depth, which a surface strip might miss.
One final thought from the study, the collaboration. Seeing so many institutions come together for a retrospective study in Epilepsia Open really speaks to the maturity of the RNS technology. We are moving past the does it work phase into the how do we optimize it phase.
Exactly. Optimization is the key word. Using sEEG to guide RNS is optimization in its purest form. It's taking the data we already have and using it to improve patient outcomes.
That's a great place to wrap up. The paper is sEEG-Guided Responsive Neurostimulation to Treat Neocortical Epilepsy, a multicenter look at efficacy and safety, a must-read for anyone doing epilepsy surgery.
Thanks, Ilara, and thanks to our listeners. We'll be back next time with more updates from the cutting edge of neurosurgery.
See you then.